FRET analysis works on the principle of overlapping emission spectra and absorption spectra of two different proteins, or these proteins are basically your fluorophores. So they are particular chemical compounds that have the ability to do this, like absorbing some sort of spectra and also emitting certain wavelengths. So here you create two fluorophores in a way that one is a bait and the second one is a prey. And FRET is going to see whether we can transfer the resonance energy from one bait protein to the prey protein in such a way that your absorption spectra and emission spectra can overlap and help to visualize secondary effects after you have given certain input. So let's begin. Basically, you try to excite your bait protein after exciting it, after sending it UV light. So your bait protein will try to emit short wavelengths. The prey protein, if it is very close to the bait, will be excited by this light and it will emit a longer wavelength. So they need to be in close proximity and or need to be bound to each other in order to express the secondary wavelength that we want to see. A similar system called bioluminescence resonance energy transfer, or BRET, is also very similar to FRET, and the basis of the BRET analysis is that if two proteins interact and their position is such that the donor and acceptor are within a distance less than 10 nanometers, then the emission spectra of one protein can be absorbed by the second protein, and hence they will be able to create luminescence of the binding protein, like the two halves of the protein that they're attached to. So, for example, there might be a BRET experiment where one protein is able to emit a light at 395 nanometers, which can be captured by the second protein, say a green fluorescent protein, and then it can emit a green light at 510 nanometer. And this ratio of light emitted by protein A to the green fluorescent protein can be measured in the BRET machine or whatever machine is going to detect these signals. We can try to purify our proteins using certain techniques. The most primary one is the affinity purification, in which we try to create a gene targeting cassette or perhaps an expression vector, which can be transformed in some organism, say an E. coli or a bacteria, and then we express it over there and perform cell fractionation so that we can extract those proteins. And then we do a process of affinity binding. This can occur on nanobeads that have exposed antibodies for certain protein targets. So it will help us to pull down or like attract all the necessary proteins to towards the nanobeads. And then we can elute the system out using magnets, because nanobeads are magnetic in nature. And we can perform an SDS page on the products of our elute. So we can see the proteins that are present. We can tag them in the process of binding to the antibodies. So this can help us to go to the next stage of purification, which is also called affinity purification, but tagged. So it is tandem affinity purification. And so here what we do is that we do an affinity column 1, where we cleave off one part of the tag by binding to a nanobead which is specific to one part of the tag. Then we have another affinity column 2, which can be cleaved off from the second nanobead attraction. So we have, for example, an immunoglobulin G bead and a calmodulin bead. And the tag which is present on these proteins will be cleaved off subsequently. So by the end of this process, we'll be receiving highly purified proteins. We can also perform even more studies on the bound or tagged proteins in such a way that we can understand or try to understand the inter-residue interactions. So for this, we can, for example, treat the tagged protein with some sort of a chemical which allows us to see the cross-linking microspectry results, which helps us to identify cross-links, or in other words, the intermolecular interactions between them.